All right, this is part 2 of the genetics lecture. I am not able to show you how to do a dihybrid cross using this computer system that I've got because I'm not dexterous enough with my fingers to draw on it. But you can watch this lecture by Mr. Anderson. If you start at 6 minutes and 47 seconds, he'll show you how to do a dihybrid cross. So if you're part of the classes that don't know how to do that already, go ahead and watch this, please. Moving on, variations on Mendelian genetics. So far, we've been discussing traits that exhibit complete dominance, where one allele is completely dominant over the other. Take this example of pea color. We can't tell just by looking at this pea. Whoa! Okay, pointer options. Head, there we go. We cannot tell just by looking at this green pea whether it's homozygous dominant or heterozygous, because the trait exhibits complete dominance. It is completely dominant over yellow. But things aren't always that simple. We also have been discussing traits that are controlled only by a single gene. Traits like this aren't actually that common. Humans have around 25,000 genes, and out of those, only about 600 are single gene traits. Even fewer exhibit complete dominance. So what else is out there? Well, sometimes one gene causes more than one trait. A great example of pleiotropy in humans is phenylketonuria, abbreviated PKU. The PKU gene is found on chromosome 12. It codes for an enzyme that converts one amino acid to another, specifically phenylalanine to tyrosine. If this gene is mutated, that causes the enzyme to be deficient. This leads to the disorder PKU. Symptoms of PKU include mental retardation, eczema, and melanin dysfunction that creates a light skin color. Because one gene affects all of these traits, it's a great example of pleiotropy. The reverse can also be true. When multiple genes work together to influence one single trait, we call that polygenic inheritance. We've been using eye color as an example of a Mendelian trait, but in fact, it's a polygenic trait. Other polygenic traits include skin color, height, and weight. Sometimes two characteristics always show up together in an organism. If you see one, you see the other. This means they are on the same chromosome. They are called linked genes. An example of linked genes includes hair and eye color, which explains why certain hair and eye colors tend to be inherited together. Blonde hair often goes with blue eyes, and individuals with brown or black hair often have brown eyes. Sex-linked genes are those found on a chromosome, a sex chromosome. In humans, sex-linked genes are typically found on the X chromosome because it's larger and has more genes than the Y chromosome. Here are some examples of X-linked traits. In fact, most sex-linked traits affect males because look at this. Males only have one copy of the X chromosome. So if they get a mutation of any one of these genes, they're stuck. They have the condition. Females, on the other hand, get two X chromosomes. So if one of their X chromosomes has a mutation, that's okay. They still have a second chance to get a functional gene. The most typical example is red-green color blindness. There are 4 kinds of red-green color blindness, but all of them make red and green look similar. Here I've got a bunch of dots with a number in the circle. Maybe you can guess the number, and maybe not. Gene-environment interaction is the situation where subtle differences in one person's genes can cause them to respond differently than another person to the same environmental factors. Because of this, some people may develop a disease after being exposed to some environmental factor, while others may not. A great example of this is bladder cancer. Individuals who smoke are at least 3 times more likely to get bladder cancer as non-smokers, but not all individuals who smoke end up developing bladder cancer. Why is this? Well, it involves a gene called NAT2. There are several versions of this gene, and smokers with one particular version of this gene have a much higher risk of getting bladder cancer than smokers with a different version. Epigenetics is the study of how behavior and environmental changes affect how genes work without changing the genes themselves. Children inherit approximately 23,000 genes from their parents, but not every gene does what it was designed to do. Research shows that experiences and environmental factors leave a chemical quote-unquote signature on genes that determines when and how the genes are expressed. Collectively, these signatures are called the epigenome. I'm not going to spend a lot of time on epigenetics because we'll have an entire lecture later on in the semester about how it works. Basically, certain activities and environmental factors trigger chemical changes that cause genes to be able to be used by the cell, and others trigger closure of those genes. Some genes display what's called incomplete dominance, where the heterozygote phenotype is intermediate between the dominant and recessive. In this example, red is dominant to white, but if a homozygous, if a homozygous dominant snapdragon and a homozygous recessive snapdragon are crossed, the resulting heterozygote is pink. Red is incompletely dominant. This is your next clicker question. You should take note of this. It may or may not be on the quiz at the end. And the correct answer here is B. Some genes display codominance, in which both alleles are dominant together. In this example, the homozygous dominant is red, the homozygous recessive is white, and the heterozygote is red and white. It is not pink. Pink would be incomplete dominance. Both the dominant and recessive allele are fully expressed at the same time. You see both of them. And this brings us to the ABO blood type system. This is an example of codominance. The ABO blood type system is how we describe human blood types. Sometimes genes will have more than 2 alleles. In blood types, we have 3 alleles, A, B, and O. Different combinations of those alleles will produce different phenotypes, including A, B, and O. So a person can have any one of these 4 phenotypes depending on which alleles they have. Letters of the alleles, A, B, and O, each refer to a different surface carbohydrate, also known as an antigen. For our purposes, an antigen is a very specific object that sticks up out of the surface of something else and acts as an identifier or name tag for that something else, so to speak. So for example, these are antigens on the surface of a bacteria. They're shapes that stick up out of the bacterium. In our example of the ABO blood type system, the antigens are carbohydrate molecules, right here. They stick up out of the surface of red blood cells. Different surface carbohydrate antigens designate a cell as a specific blood type. For example, here we have this antigen. This is the A antigen. And if a red blood cell has this antigen sticking out of its surface, we say that it is a type A blood cell. The same for type B. This is the type B antigen. If a red blood cell has this antigen sticking out of its surface, we say it is a type B blood cell. And type O blood has no antigens whatsoever. Lots and lots of living things have antigens on their surface. Some antigens are proteins, some are carbohydrates. They can be all different kinds of things. The great thing about antigens in general is that they allow the body to distinguish between foreign intruders and things that are safe. They act as name tags, so to speak. If an antibody is flowing along in the bloodstream and it bumps against an antigen, it will recognize that antigen by its shape as being either part of the body or being a foreign object that must be destroyed. If the antigen shape indicates that the object is foreign, the antibody will bind to it and alert the white blood cells that it needs to be destroyed. But let's get back to red blood cells. Red blood cells can either have antigen A on their surface, in which case they are blood type A. They can have antigen B on their surface, in which case they are blood type B. They can have both antigens A and B on their surface, making them blood type AB. Or they could have no antigens at all, making them blood type 0. It is very important to note that blood type alleles code for the antigens. If a person's DNA has allele A, then that calls for the A antigen, and the red blood cell will display the A antigen, and the person will have the blood type A. If a person's DNA has allele B, their red blood cells will display the B antigen, and the person will have blood type B. If the person's DNA has allele O, then their cells will not have any surface antigens at all, and the person will have blood type O. Here's a clicker question. Go ahead, pause the video and answer the question. And the correct answer here is C. So let's fill out this table for phenotype and genotype. If a red blood cell has the A antigen, the phenotype is type A. Here we go. What are the possible genotypes? Remember that when we're talking about genotypes, we're talking about alleles. And all the alleles we have for the ABO blood system are A, B, and O. So which combinations of alleles will give us type A blood? Well, we know there's at least one A allele because it's type A. Could the other allele be A? Yes. AA is a possible genotype. Could the other allele be O? Yes. Since recall that the allele O does not call for any antigen. AO is a possible genotype as well. Since O calls for no antigen and A calls for the A antigen, all we have is the A antigen on the red blood cell, and that means it is type A. Could the other allele be B? No, because that allele calls for the B antigen, and we don't see that here on type A blood. Let's keep going. For antigen B, the phenotype is type B, and the possible genotypes, we've got to have a B allele, so the second allele could be another B, or the second allele could be O, just like with type A. Antigens A and B, you have type AB blood, and the alleles are allele A and allele B. And if you have no antigens, you have type O blood, and your alleles are going to be O, O. Back to antibodies. Antibodies are found in the bloodstream, floating around and mixing with the red blood cells. Each antibody fits the shape of a specific foreign antigen. If that antibody happens to bump into its corresponding antigen, it binds that antigen and alerts the white blood cells that an intruder has been found and needs to be destroyed. It's very important to remember that each type of antibody is a specific shape designed to fit one specific antigen. Here we see some antigens on the surface of a bacterium. We have a square antigen and we have a Pac-Man shape antigen. We see two different antibodies. This one is anti-square and this one is anti-Pac-Man. That's funny, anti-Pac-Man. The anti-square antibody cannot bind to the Pac-Man antigen because it is the wrong shape. And the anti-Pac-Man antibody cannot bind to the square antigen because it is the wrong shape. Here is another clicker question. Pause the video and answer. The correct answer is B. When antibodies encounter something they fit, they grab onto that thing and clump together. This is called agglutination. Here we have an image of agglutination, and this makes it easier for the white blood cell to swallow up the intruding germ or whatever has alerted the antibodies. If compatible blood types are not matched during a blood transfusion, the antibodies in the recipient, right there, will attack the donor blood cells, thinking they're a foreign agent that shouldn't be in the body, and that is agglutination. On top is a microscope image of normal red blood cells, and on bottom shows agglutination, what happens when incompatible blood types are mixed. Agglutination causes blood in the urine and acute kidney failure, among many other things. Here we've got an image of the blood of someone with type A blood. This person makes anti-B antibodies that search for the foreign B antigen, and when it sees the B antigen, it binds to that and starts agglutination. This is why a person with type A blood cannot get a transfusion from someone with type B blood. Now let's go back and fill in the antibodies in the plasma column. Plasma is another part of blood, by the way. So if a red blood cell has antigen A, it's going to produce anti-B antibodies. And if it has antigen B, it will produce anti-A. And if it has antigens A and B, it will not produce antibodies. And if it has type O, it will produce both anti-A and anti-B. Let's think about this. A type O person has what antibodies? Looking at the chart, we see that they have anti-A and anti-B. So pause the video, answer the question. This person cannot receive type B blood. Here is another one. A man and a woman both are heterozygous for type A blood. What genotypes can their children have, and what genotypes can their children not have? Pay close attention. This may or may not be on the quiz at the end. So if they are heterozygous, that means the alleles are different. So they have one A and one O. And it says that they each are the same. So let's do a Punnett square. So here we're going to have AA, we're going to have AO, AO, and OO. All right, let's look at letter A. What genotypes can their children have? Their genotypes, the children can have any of the genotypes that are in this box. So they can have AA, AO, AO, or OO. What genotypes can their children not have? Well, their children cannot have type AB, or type BB, or type BO. Basically anything at all with a B in it, their children cannot have. This is a concept map that you are allowed to do if you really want to. And that's it. I hope you have a lovely weekend or week, depending on, where you are in the week. That's all.